Good afternoon. I'm Charlie Roberts. I'm director of the Comprehensive Cancer Center here at St. Jude Children's Research Hospital. And with my co-host, Dr. Charles Mulligan, it's our pleasure to welcome you to the inaugural lecture in the Science of Childhood Cancer series. Before we launch our first lecture, I want to take 2 minutes to virtually introduce you to St. Jude Children's Research Hospital. While many know us as a cancer hospital, not all realize the broad scope of our scientific endeavors. This is our campus in Memphis, Tennessee, which is 90 acres with 30 buildings and over 5,000 employees. Science has always been at the heart of our mission to advance cures for pediatric cancer and other catastrophic diseases of childhood. Our unique funding model enables exceptional support for research, and our endeavors span the full spectrum from fundamental basic science, as reflected by work leveraging 2 cryo-electron microscopes and the world's most powerful NMR spectrometer, to translational research that brings advances to patients, to our clinical research in which 60% of our patients are on therapeutic trials, and to our survivorship research that includes 2 of the world's largest survivorship studies. Central to our mission is our commitment to share data and work collaboratively with researchers around the world to most rapidly advance discovery and cures for children. You can see many of those free resources at stjude.cloud. In furtherance of this mission, we are delighted to today launch the Science of Childhood Cancer lecture series. It is our goal over the next 15 weeks to provide a platform for cutting-edge science from leading experts at top institutions whose research is relevant to better understanding the mechanisms that underlie cancer, to developing better treatments, and to enabling a better quality of life for survivors of childhood cancer. It is great to see so much interest in this series with over 2,000 registrants, including investigators from around the globe. And today we launch with the exceptional science of Dr. Michelle Sadelain from the Memorial Sloan Kettering Cancer Center. Some quick housekeeping. If you have a question, please type it into the chat and send to all panelists. There are also emails for you to send questions and emails if you have any technical needs. With that, I'm going to turn it over to introductions by Dr. Doug Green, who is co-leader of our cancer biology program and who is St. Jude's most recent inductee into the National Academy of Sciences.
Hello, everybody. I'm Doug Green. I'm the chair of immunology at St. Jude, and it is my distinct pleasure today to introduce Michelle Sadelain. Michelle is currently the director of the Center for Cell Engineering and holds the Stephen and Barbara Friedman Chair at Memorial Sloan Kettering Cancer Center, where he's a member of the immunology program and the departments of medicine and pediatrics. Michel has his MD from Paris, from the University of Paris, and his PhD from the University of Alberta in Edmonton, Alberta, Canada. I met Michel 35 years ago as a graduate student, and it was my privilege to co-supervise his thesis with Tom Wegman, a wonderful immunologist from Boston and was living in Canada, working in Canada. Even as a graduate student, Michel had a vision that he was going to engineer T lymphocytes to treat cancer. Now, I'm going to be honest, when he posed that back in the day, remember, this was 1985, where we were working with stone knives and bearskins. Michel suggested that and was committed to the idea, which we thought was a little far-fetched. A mere 25 years later, after having worked with Richard Mulligan on retroviral gene transfer, studying stem cells and transduction of stem cells, Michel's team was the first to treat BALL cancer with CD19 CARs successfully. It's a stunning achievement. We're all thrilled with the results, and we're all well aware of the importance of this incredible advance. So with that introduction, I want to introduce my friend, Dr. Michelle Sadelain.
Thank you, Doug. It's hard for me to speak after an introduction like that. First, I want to thank you and Charlie for the honor of participating in this series. It is a true pleasure to always serve in any way for St. Jude Children's Research Hospital. We all admire the mission, the quality of the clinical care, and the outstanding research that takes place there. I'm also pleased to talk to some of my colleagues at St. Jude who have made important contributions and continue to do so in the field that I will be discussing today, the field of CAR T cells and CAR therapy. And lastly, Doug, it gives me again immense pleasure to see you and to be introduced by one of my dearest mentors. The structure of my presentation is somewhat historical. I thought that for those not so familiar with immunology or immunotherapy, it could be of interest to run the course of history, going back in time, as you'll see, but then, of course, addressing the present and the future of this emerging field.
And Michelle, I'm going to step in here just for a moment and remind people that we are going to invite questions. As you think of questions during Michelle's seminar, type them into the chat, and I will try to bring as many of those questions up when Michelle pauses for questions.
Thank you, Doug. Well, T lymphocytes are at the center of this talk today, and they are born in the thymus, where they acquire the letter T and also a receptor for antigen, a unique receptor known as the T cell receptor, which is clonotypic, that is to say, characteristic of every T cell that forms such a receptor. This receptor is formed through the recombination of a number of genes called VDJ genes that recombine and in theory can generate up to 10 to the 18 different specificities. Now, those who have attempted to quantify the size of what is called the human T cell repertoire come up with a number of about 20 million different T cells that an average human being would harbor, which gives us a chance to respond to a large number of antigens, although perhaps not all of them. And so in the thymus, these T cells are formed, blindly creating these receptors, which are then edited or pruned, restricting this repertoire that must be tolerant of self, and yet capable of recognizing as much as possible. Now, even before T cells had been identified in the 60s by Jacques Miller, before that, immunologists had already developed a technique known as adoptive transfer of cells. And so from an immunized mouse or an immune-tolerant mouse, they would isolate circulating cells or lymph node cells and transfer them to a syngeneic animal and study its impact on the immune response. As early as the 80s, this approach inspired some clinicians to undertake what was then called an adoptive transfer of T cells and adoptive T cell therapy. And those started in part somewhat accidentally in the course of bone marrow transplantation. I say accidentally when at the time of the first transplants, it wasn't appreciated that a bone marrow transplant is not only the transfer of hematopoietic progenitor and stem cells, but also of T cells, and then evolved into a series of different strategies listed here to the right: donor leukocyte infusion, the use of lymphokine-activated killer cells, tumor-infiltrating lymphocytes, and also virus-specific T cells. Now, don't think that T cells were immediately accepted and embraced as therapeutic agents. In fact, in the upper left here, I show a very impactful paper from the late 70s that in mouse models, in so-called nude mice, showed that if you induced tumors with chemicals, there was no difference. There was no difference in the induction of those tumors, suggesting that, as the authors concluded, that there is no active role for thymus-dependent immunity in surveilling tumor development. And so a number of people formed the impression that T cells don't really matter in cancer development or cancer therapy. At about the same time, Rolf Kissling and colleagues in Stockholm uncovered another immune cell type called a natural killer cell. And soon after that, it became apparent that, in fact, these nude mice do have some residual dysfunctional T cells and also many of those NK cells that could account, in fact, for immune resistance to cancer. And it then took many years to rehabilitate, if you like, the role of T cells in cancer, something to which Bob Schreiber and Lloyd Old in particular contributed greatly. And so this covers this sort of early part of T-cell-based medicines and T-cell therapies, with the gradual notion arising from a variety of approaches that T-cells can be effective, they can be dangerous and sometimes cause lethal graft versus host disease, but that one has to enrich these various sources of cells in the right T-cells, those that recognize the tumor antigen. And so then comes the period that Doug already alluded to, actually in the mid-'80s. And as he already said, Doug was on my PhD committee at the time. I formed this idea that perhaps you could engineer T cells to instruct them, to acquire any specificity of your choosing, and possibly direct them against cancer or other pathogens. Now, that would first require a genetic construction, a genetic modification of the clonotypic T cell, introducing into it a novel specificity. And so that required to, for me, in any case, to learn more about gene transfer. And so I went to MIT to learn what were then the rudiments of gene transfer biology in mammalian cells. David Baltimore, Howard Temin here had already unraveled one of the mysteries of retroviruses, the reverse transcriptase enzyme that reverse transcribes RNA genomes into DNA, leading then to their integration and therefore a stable genetic modification into mammalian cells. And Dusty Miller and Richard Mulligan, by the late 80s, were developing what's called packaging cell lines, which would allow to produce replication defective recombinant retroviral genomes that could be used for the purpose of gene transfer. So transferring genes into T cells wasn't immediately evident. In fact, in this slide, it took me, I recall, 3 years of work until I could introduce a few copies of the E. coli LAC-Z gene into T cells using a recombinant murine leukemia virus-derived retroviral vector. Today, we can teach a high school student in an afternoon to introduce genes into T cells, even several genes, using a variety of methods. Retroviral vectors, as you know, I think, for the most of you, are still today the tool of choice for the generation and production of targeted T cells. So one needed to redirect the specificity of the T cells towards an antigen of one's choosing. And here the story begins, I think, with the cloning of one of the chains that makes up this, what's called the CD3 complex. T cells recognize HLA peptide complexes through a dimer of an alpha and a beta chain, which confer the specificity to that T cell. But the activation is mediated by a complex called CD3 made up of multiple chains, including one called the zeta chain, which is shown in the upper left here in green. When three groups independently cloned this chain, Brian Seed, Art Weiss, and Rick Klausner, they all created these fusions that you see here in panel A. The fusions were created because this chain lacks an extracellular domain, and at the time they weren't sure whether they had cloned the full cDNA. And so with these recombinant zeta chains, they established, by cross-linking them with antibody and Jurkat cells, that the zeta chain was capable of initiating T cell activation. And then two investigators creating what's shown in panel B here. And that is swapping those extracellular domains for SCFVs, that is to say, the heavy and light chains derived from immunoglobulins, to confer in principle to that zeta chain any potential specificity. And in recognizing an antigen through an SCFV, such a molecule would not require HLA to engage antigen, because it is not recognizing an HLA peptide molecule, but rather any molecule present at the surface of a target cell. And so both Zeliger and Thomas Broecker in 1993 described such receptors. Because we had the ability to introduce molecules not just in clones or hybridomas with primary T cells, we soon realized that these chains were capable of directing killing by T cells, but struggled to allow for the T cell to divide, to increase in numbers, and to retain its function, at least for a certain amount of time. And as you will hear soon, that is very critical to the engineering of therapeutic T cells. And so having observed that this zeta chain fusion was incapable of directing the amplification of T cells, we started looking at co-stimulation and engineered chimeric co-stimulatory receptors. As shown here, the first one was based on this CD28 co-stimulatory receptor that I'll discuss further a bit later. That is a essential receptor that assists the T cell receptor in producing productive immune responses. And having shown that we could create such fusion receptors, we then merged them, creating a dual signaling molecule that we then called the chimeric antigen receptor. And by deference to the first zeta chains, we called it a second generation chimeric antigen receptor. And this molecule could therefore recognize a cell surface antigen, initiate activation, including cytolytic activity, and also through its costimulatory domain, allow for the T cell to divide and amplify in numbers upon repeated exposure to antigen. Now, one needed to work with a target, and there were several targets already in this small emerging literature. We sought one that we thought we could someday bring to the clinic and turned our eyes towards leukemias and lymphomas, thinking that that would be perhaps the type of cancer in which we would first attempt to bring this therapy to the clinic. And we set our eyes on CD19, CD20, and CD22, three molecules already well known in the mid-90s, known to be expressed in B cells, as well as the vast majority of lymphomas and leukemias that arise from the B cell lineage. And back in 2003, we provided this first demonstration that you could take collect human peripheral blood T cells, transduce them with a gamma retroviral vector encoding for a CAR specific for CD19, and following a single infusion of these T cells, induce these durable remissions with more T cells that could actually even induce a remission in all of these mice and in fact cure these mice. And so we enter here now the second period in the present, the time of CD19 CAR therapy. Now, to bring this to the clinic, we needed something very critical, which is the ability to manufacture these T cells at clinical grade, satisfying the requirements of the Food and Drug Administration. And here I would like to pay tribute to a few people that I refer to in this slide as key contributors to the manufacturing of T cells of clinical grade. In the upper left there, I want to refer to Bruce Levine and Carl Jude, who had developed a system of iron beads coated with antibodies directed to CD3 and CD28 that we, and many others have used for the production of T cells, including later CAR T cells. I want to mention Bruce Levine at the University of Pennsylvania, who had pioneered the production of lentiviral-mediated gene transfer into T cells, working with Virexis some 15 years ago, working with that company to develop an HIV therapy using antisense sequences. I also want to mention Mark Dudley from the National Cancer Institute, who greatly assisted Steve Rosenberg and his team to develop their genetically modified T cell program. And lastly, I want to salute my colleague, Isabelle Rivière at MSK, who established the process seen in the upper right, where essentially you start from an apheresis product from the cancer patient, then activate the T cells using those activating beads or other systems, and then transduce these cells, and in this case, and shown in the lower right, in these bags that you see here. And after a few days, remove the iron beads and QC test these cells, run some quality control tests, and then either infuse those cells or freeze them down for deferred use. And with the ability to manufacture T cells and then CAR T cells, in fact, these three centers then went on to pioneer CD19 CAR therapy. In fact, the NCI was the first to report a case report in a patient with non-Hodgkin lymphoma, an adult with relapsed and refractory disease, showing a response after an infusion of CD19 CAR T cells. The next was Carl Jude at UPenn, who reported on 3 patients with chronic lymphocytic leukemia, CLL, in whom, again, he obtained a very dramatic complete response. And then, as shown here on the left, our own study in the first 5 patients with relapsed refractory ALL, we focused at MSK on adult patients. Again, all 5 of them going into a complete remission with undetectable circulating leukemia cells within 2 weeks of the T-cell infusion. And in the marrow, as shown here in these FACS profiles, the complete ablation of any normal B cell, because the CAR T cell cannot distinguish a normal B cell from a leukemia, or leukemia, sometimes as early as 11 or 12 days after T cell infusion. These were molecular responses with undetectable tumor by PCR or deep sequencing. Of course, we were thrilled when later that year, the Science Journal declared cancer immunotherapy to be the scientific breakthrough of the year. Much of that was for checkpoint blockade, the first big immunotherapy, which I will come back to later in this lecture, but also for CAR therapy. And as you know, these early results were then confirmed at those 3 centers and at many others, thus generating immediate acceptance, if you like, of these results as being very real and reproducible in the hands of different groups. It's around that time, in an interview by Denise Grady, asking about what exactly is this form of medicine that doesn't use a chemical or an antibody, that this answer was given. What we do are creating is to create living drugs, genetically engineered targeted T cells that are released in the patient, can undergo expansion, and ideally, anyway, would keep fighting the cancer for as long as there's cancer to be found. And so these are the 2 molecules that were approved by the Food and Drug Administration in 2017. Both of them are 2nd-generation CARs, and both of them are specific for CD19. One of them utilizes the costimulatory domain of CD28, and the other, to the right, uses the costimulatory domain of 41BB. And here's my first salute to a key contribution from St. Jude. This was the work of Dario Campana, together with Terence Geiger, who today continues to advance and promote CAR therapy at St. Jude. So these 2 molecules are the first to be approved, the prototypes, if you like, for the field. And before moving any further, I will, well, I just want to say one more thing. This spurred tremendous interest in this approach. And as of today, according to Nature Biotechnology, there are over 500 clinical trials worldwide making use of CAR T cells, many continuing to explore CD19 as a target, and also many now starting to explore this approach for other cancers. And as shown here on the right, there are 2 countries that really largely dominate, if you like, by the numbers, this field, and those are the US and China. In fact, China has now surpassed the US in the number of CAR trials. So before proceeding any further, I would like to ask Doug if there are any questions at this time, addressing sort of this history, if you like, of CAR therapy.
Yes, thank you, Michelle. We have a question from Sarah Luling, who asks, what are the effects of CAR T cell therapy on the normal functions of the immune system? Is it weakened, strengthened, or does it continue to function normally?
Okay. Well, maybe I should, I'll answer this question after my next few slides. after presenting sort of the state of the art in just 2 slides
Okay.
in the patients. And then I will make sure to address that question.
If you don't, I'll bring it back to you. Another question is one that I have. We know that there is a great deal of work that's been done on creating the chimeric portion of a CAR, the signaling portion, and a great deal of work is ongoing to identify targets, target proteins for the specificity. What do we know about the selection of the antibody? If you have 10 antibodies that all recognize CD19, will all of them be equivalent in terms of the FV contribution to the CAR, or are there considerations of affinity, fine aspects of the specificity, et cetera?
Yes. So selecting a good SCFV is obviously important. It has to be specific for your target, not cross-react by chance to any other molecule expressed on a normal cell type and ideally in any other organ. So of course, it's specificity is key. The role of affinity is not fully unraveled yet. Most of the cars that were built over the past decade made use of heavy and light chains derived from high affinity antibodies. I think that on the one hand, the assumption was high affinity should be good. And on the other, it's practical, as most of the hybridomas that were available for cloning and extracting the heavy and light chains were generated by scientists who were purposefully looking for high affinity antibodies. And so the field really started with high affinity CARs. But as we explore them today, we start seeing that high affinity is not an absolute requirement. If you start, so most CARs are in the nanomolar range, but if you go down to the micromolar range, you start having CARs that don't work anymore. Now, in between, what is the optimum? I don't think that the rules are yet known. One has to balance the ability to engage antigen and initiate activation, the risk of antigen escape if the number of ligands of antigens is very low, but also the risk of T-cell exhaustion or even activation-induced cell death could be if the signal of the activating signal were too strong. And so I think that there's still a lot to be learned with regards to that.
There are additional questions, Michelle, but I will hold those until your next pause.
Okay, my next pause will be, I think, near the end at this point. So how to describe the state of the current state of CD19 CAR therapy very briefly in 2 slides. Well, here are 4 large studies that were reported in 2018. The 2 on the left make use of a CD28-based CAR specific for CD19. The 2 on the right of a 4-1BB-based CAR. So the upper left is an update on the first 53 patients that we had treated at Sloan Kettering, who were, again, adults with refractory and relapsed disease. The complete remission rate was high, on the order of 85%. But what you see with this survival curve is that over half of them will eventually relapse and die from their disease. And so the good news is this very stable tail that you see. But the first 18, 20 months there show you that a number of patients will eventually relapse, especially those with higher tumor burdens. So there is room for improvement. And the upper right is the study in children from CHOP, reported by Shannon Mode and colleagues. And you'll see here the x-axis is shorter, but this was their first 75 patients. So going out 22 months, but you see here too, very high complete remission rates, again, on the order of 85 to 90%. But then about half of the patients, perhaps a bit less than in adults, will eventually relapse. So again, excellent news to see patients who had no other therapeutic option to at least have an extended survival, but clearly room for improvement. And the two lower slides are non-Hodgkin lymphomas. One, again, is a multi-center study using a CD28-based CAR on the left or a 4-1BB CAR based on the right. And again, in a nutshell, the complete remission rate in these patients with refractory disease is a bit less than in leukemia, actually. But on the other hand, the durability is a bit greater than in leukemia, although here too, not too far from half of patients will eventually relapse. So there is room for improvement. So we take great interest in the toxicities and the failures of this therapy, because that is where we will learn how to make it better. And so the two toxicities that are highlighted here were two toxicities that were not anticipated from the original mouse models. And one has now been named cytokine release syndrome. It's characterized by fevers, persisting fevers, hypotension, and eventually hypoxia. There is now a gradation of this toxicity and improving means to control it. First, to recognize it early on and to control it. The second one is a neurotoxicity, which in the beginning was very puzzling and very surprising to the clinicians taking care of these patients, characterized by global encephalopathy or clonus or seizures, or sometimes a spectacular aphasias, also reversible, which now has been named ICANS, and for which there is also a classification and guidelines for intervention. I'll just say one word about the CRS. It took us a while to model it in mice. Surprisingly, it occurs quite frequently in patients, especially those with high tumor burden, but it was so hard to model in mice. And from those studies, we confirmed really that much of the cytokine response that is toxic, if you like, to the patient, in particular the production of interleukin-6, comes not from the CAR T cells themselves, but rather myeloid cells in the recipient. And so these CAR T cells not only engage and destroy tumor cells, but they activate other cells in the recipient, and in particular the myeloid compartment. And that is the cause of much of the acute toxicity. It also raises an interesting question of how one could further engage the myeloid compartment to further fight the cancer. To the right are mechanisms of resistance, if you like, failures of the therapy, and they fall in three main buckets, if I may say. The first is are instances where the quality of the T cells can be implicated in the failure. And that's really through the development, a loss of function in the T cell, the development of T cell exhaustion. There are also factors that can be attributed to the microenvironment. And like for any other immunotherapy, a number of barriers, and the three that are highlighted here are well-known barriers to immune responses in the tumor microenvironment. Those three have been shown to indeed downregulate or even inhibit the function of CAR T cells. A third one has to do with antigen escape. And because you target one antigen, the expression of that antigen on the tumor cells is very critical. You want to ideally have an antigen found on all tumor cells and at a level that is sufficient to trigger a strong CAR T cell response. But when there is complete loss of antigen, obviously this is material for relapse, and this has been seen and reported in many patients. Another interesting category of antigen relapse, which I won't have time to go into today, are instances where the tumor comes back, relapses, but with very low level expression of the antigen. And so the question there is, well, why is the CAR not eliminating those cells, since in this case the target is still there, and there's much to be learned about that today. So a question that was about the long-term the effects of the CAR T cell therapy, well, they are complex and not fully appreciated yet. There are these acute events, and in particular the cytokine release syndrome. The physiopathology of the neurotoxicity is not well known. The cytokines contributed to it. There is microglial activation, but there's also damage done to the blood-brain barrier, and the mechanism for that is still not well understood. As for long-term consequences, well, there isn't so much information yet, but some studies have noted that perhaps there's an increase in the recurrence of some viral infections in the long term. And if that's the case, well, it certainly means that this should be further investigated in the clinic, again, for the long-term follow-up of these patients. So I'd like to turn to the future now and discuss some new directions in the field of CAR T-cell engineering, addressing the advent of gene editing as a new technology to engineer T cells and to engineer better T cells, but other aspects as well. So let's go back to these two foundational CARs shown here on the left again, both specific for CD19, but one making use of the CD28 costimulatory receptor and the other one the 4-1BB receptor. And so here we introduce them in mice that bear a CD19 positive leukemia purposefully at relatively low T cell doses. And we compare them side by side. And on day 7, we can see that there's a similar number of those second generation CAR T cells in the bone marrow, which are much more abundant than that first generation, the zeta chain based CAR that you see here on the left, which is unable to expand and persist in these mice. But note the effect on the tumor. The 28Z CAR T cells have essentially already eradicated leukemia in these first 7 days, but not the 4-1BB CAR. And I remind you that their T cell numbers are very similar at this time point. And so this fits very well with other data that we and others, several others, have reported, that the CD28-based CAR T cell is a more potent effector on a cell-per-cell basis. However, this CD20 CAR T cell doesn't persist very long, as you see here, in contrast to the 4-1BB CAR T cell. And through its greater persistence, it too eliminates the tumor. It just takes a little longer. I insist on this because it mirrors clinical observations. And so it is a very believable, if you like, result. And that really confirms this key difference between the two CAR designs. As you saw from my very rapid summary of clinical activities, the two are very comparable overall in the remission rates that they induce in patients with different B-cell malignancies. But remarkably, they do it with very different kinetics. And so if you wanted to improve these CARs, you would probably say, well, I could make a 4-1BB CAR T cell that's a more potent effector, or I could design a CD28 CAR T cell that persists longer. And we chose to do the latter. And here we formed the hypothesis that perhaps this CD28 and CD3 zeta assembly in the CAR design was overstimulating the T cell. On the left here, you see the T cell's receptor CD3 complex with its zeta chain, and separately on the left, the CD28 costimulatory receptor. These costimulatory receptors migrate into the synapse when the T cell engages its target HLA peptide complex. And so the timing and the stoichiometry is not that of the CAR, where there's a covalent bond, a 1-to-1 fixed stoichiometry. And because of the similarity of the CD3 zeta chain signal and the CD28 signal, we thought this might be excessive, accounting for sure very good effector function, but also perhaps abrogating the persistence of these T cells by exhausting them too fast. And so we explored the zeta chain component and created these mutants. These activating motifs, there are 3 in the zeta chain, have been very well defined by immunologists for over 20 years in mouse models, but not in CARs. And so if we compare in this mouse model a CAR, the CD28 zeta wild type CAR, to these mutants, we find that some are worse and some are better. So again, at this particular dose here of CAR T cells, the 28Z, this foundational CAR, again, FDA approved for some indications, at this dose only induces some delay in tumor progression and a few long-term remissions. If the CAR only retains the third ITAM, you can see there's a transient response and all mice relapse. If, on the other hand, it retains the proximal ITAM, ITAM1, you can see that there's tumor progression because of the low dose of T cells in the beginning, and then the T cells catch up to the leukemia and eliminate it. I don't have time to review this published work, but suffice it to say that we found that when we introduce these variant CARs, this 1XX or the XX3 CARs, into human peripheral blood T cells, including naive T cells, these CARs really can direct the differentiation of these cells, in addition to their effector function and also to their tendency to exhaust rather soon or exhaust rather later. And so we believe that a diminished strength of activation in the context of a CD28-based CAR is a very favorable feature. And in fact, I'm pleased to say that COVID permitting, by September or October, we plan to treat the first patients at MSK using a 1XX-based CAR. The advent of targeted nucleases, and especially CRISPR-Cas9, made it possible for us to ask the following question. Does it make a difference where the CAR cDNA integrates into the T cell? As you know, if you use a gamma retroviral vector, a lentiviral vector, or a DNA transposon, all of those are effective in introducing the cDNA into the T cell, but they result in semi-random integration, which we know will lead to variegated expression. And so here we could ask with these tools, how much does that really matter? And pinpoint the site of integration. And one of particular interest to us is what's called the TRAC locus, the locus that encodes the alpha chain of the T cell receptor alpha. So here you see the CAR cDNA being targeted to this locus, and note that the donor DNA here for homologous recombination, encoded by an AAV6 vector, lacks a promoter, such that the CAR will be expressed under the transcriptional control of the TCR promoter. And we go back to these low-dose experiments with stress tests, and you can see the difference here. This is the conventional CAR T cell with the retroviral vector. At these doses, there is no long-term survival. And you can see that the equivalent, identical dose, but of the TRAC-edited T cell is much more effective. This control here is one where we knock out the TRAC, but still introduce the CAR using the same semi-randomly integrating retroviral vector. And you can see that there is no benefit from having knocked out the T cell receptor, which really told us that it's not getting rid of the endogenous T cell receptor that makes for a better T cell, but rather the transcriptional control that you exert on its expression. And the next slide, again, this was published, and I will not go into it any longer, is just to say that we have some insights into the mechanism for this. So the edited T cells didn't exhaust as fast, they didn't proliferate more, they didn't persist more, but they remained functional much longer. And we also noticed that the CAR, upon contact with antigen, is internalized and degraded. And that gave us an explanation for why the transcriptional control is so critical, insofar that you need to replenish with de novo translated CAR molecules at the cell surface to allow for the CAR T cell to continue to function. But we take from this that it's very helpful for the T cells to have a little break and not to be constantly in the on mode, if you like. And so gene editing is coming to the clinic. The University of Pennsylvania, Stadtmauer and colleagues, together with Carl June, just a few months ago reported a first series of 3 patients to whom they infused a gene-edited T cell. It was a triple knockout, deleting the alpha and beta chains of a T cell receptor as well as PD-1. And in this case, not introducing a CAR, but a T cell receptor using a lentiviral vector. So the approach was overall safe, although it was noted that this multiplexed editing causes quite a number of translocations in particular, and that still requires attention. And on the left here, I'm just showing the trial that we are planning that is not open yet, but hopefully will open at the beginning of next year at MSK, which is based on the study that I just showed you, where in this case, we are knocking in the CAR to a designated locus, again, the track locus. And our anticipation is that if each and every T cell is better than what you get with variegated gene expression using conventional vectors, that then you should need fewer T cells. And if you need fewer T cells, the manufacturing should be, in principle, simplified. And also perhaps that the toxicities will be reduced, because we do know that the key toxicities of CRS and ICANS usually occur in patients in whom the T cells are given at high number and undergo vast expansion. Just one last note on another project that I don't have time to go into, but we are exploring the human genome in T cells. We're looking for what's called genomic safe harbors. We're looking for places where we can introduce safely therapeutic genes that could contribute to further increase the potency and persistence of T cells. But for that, we need loci or extragenic sites that will not perturb endogenous gene expression, that do not undergo heterochromatinization and silencing, and allow for regulated expression of the transgene, which may be inducible or whatever. And so for that, we are looking and have now identified recently what we call genomic safe harbors, and we believe that those would be useful to further design better T cells. Now, where to apply CAR T cell therapy? I would like to here mention the big brother of immunotherapy, checkpoint blockade. This slide just shows, for those of you not familiar with this, the multitude of costimulatory receptors and their ligands that contribute to shaping normal T cell responses. And so nature has yielded, evolution has yielded so many molecules that allow to fine-tune, again, the function of T cells for different settings, different types of infections, etc. And as you already heard and know, using these costimulatory molecules is very pivotal in controlling the response, not just in CAR T cells through the design of second-generation CARs, but also using antibodies to activate or to de-repress T cells. And so these molecules can be activating or inhibitory. And of course, the two best known, most famous at this point, are the CTLA4 and PD-1 inhibitory costimulatory receptors, the blocking of which can enhance T cell responses in cancer patients, and for which, the Nobel Prize was given to Jim Allison and Honjo last year, or 2 years ago. So I'm just reminding you of this importance and success, formidable success of checkpoint blockade, to address here the question of where should we apply CAR therapy? Well, I think all of you are very well familiar with this graphic that plots somatic, the load of somatic mutations found in different cancers. And in gray here, I've outlined those that tend to respond well to checkpoint blockade. And the current thinking about that is that if you have more mutations, it is more likely that some of them will form what's called a neoantigen, meaning a variant of a normal protein that can be antigenic and immunogenic in the patient, if you have the repertoire to recognize such a neoantigen. And in fact, a high mutation burden is a favorable criteria for responding to checkpoint blockade therapy. But then a lot of cancers have low mutation burdens. And this is perhaps one area where pediatric cancers, in fact, may fit in this overall description, although all cancers also have some patients with a low mutational burden. In fact, the response that we saw in ALL is in one of those cancers that have a low mutation burden. AML patients present on average with only 13 mutations at diagnosis. Those are low mutation burdens, not 500 mutations or 1,000 or more, as you may see in cancers or melanomas. And so perhaps this is where CAR therapy could be the most effective. where checkpoint blockade has a lesser probability of being active. Now, for that, we need to identify targets. And we reported 3 years ago on an algorithm that we use anyway at Memorial that we developed in a project focusing on AML, starting with cell surface proteomics and taking into account transcriptomics, which are widely available. We assembled a list, and we also merged the data from the normal human protein atlases, or some of them that we selected, and started looking for proteins that would be overexpressed in AML, not found at all or at any level of concern in normal tissues. And from this, started identifying novel targets for AML. And we're now trying to expand this approach. And there's one recent example that I want to bring up, working with the laboratory of Scott Lowe at Sloan Kettering, who focuses very much on tumor suppressors, oncogenes, but also on the process of senescence. We applied this approach to look for targets in senescent cells. And so in Scott's Lowe, senescence was induced in different ways, and transcriptomics and proteomics were performed. And what was induced was compared to this normal proteome atlas. And one target in particular drew our attention. It's the gene called the PLOR, which encodes a cell surface molecule, so therefore a potential CAR target, which is the urokinase plasminogen activator receptor, UPAR. And in different mouse models, working together between the two labs, we showed that CAR T cells targeting cells induced into senescence, either through oncogene manipulations or by chemical injury or by an unhealthy diet leading to liver fibrosis, we could reverse the course of these pathologies by selectively targeting and depleting senescent cells. So I think that you can see where I'm going, that CAR therapy, I think, can play a role way beyond CD19 CAR T cells, and in fact, play a role beyond cancer. So in myeloma, there are some exciting results generated by different groups today that look promising. Solid tumors, we all know, is a big challenge. And you're very fortunate to have Stefan Gottschalk and his team working on that at St. Jude. And we think that CAR T cells could be useful in other instances, autoimmunity, severe infections, regenerative settings, including this targeting of senescent cells that I just alluded to. The last point I want to make, and Doug, I don't want to go over time, so I'll be very quick about this, is another exciting development in the field right now is that more and more immune cell types are being explored for their potential to serve as therapies for different cancers or other pathologies. And so T cells are being used, NK cells are being used, and there may be some other innate immune cell types. Macrophages are also being engineered today. And to me, that is very exciting. One aspect that I want to highlight here that is perhaps futuristic but of interest to us is the ability to generate immune cells and CAR T cells in vitro. And that is to say, not to start from a patient or a healthy donor in which the T cells were born in the thymus that I showed you in my first slide, but rather T cells that would be born in a dish or in a bioreactor. And so we provided one element of proof of principle a few years back in this study where we generated induced pluripotent stem cells from T cells, introduced a CAR, and generated in vitro lymphoid cells that could treat a not too hard to treat cancer in the mouse. And we continue to work on this, and this would warrant a much longer discussion and a whole presentation. And but we are hopeful that we will be eventually able, we're not there yet, but able to design T cells with different features that would be obtained not just by engineering a CAR, a synthetic receptor, but also by directing their differentiation to acquire, again, the properties that we want them to acquire. So the CD19 paradigm has affected the world of T-cell-based medicines in the following way. For decades, as I quickly reviewed at the beginning of this lecture, investigators focused on finding the right T-cell in a donor or in the patient himself or herself. But what I think the CD19 paradigm shows is that it is now possible to conceive of T cells as drugs, cell-based drugs, living drugs, and that perhaps building on this example, we will be able to ultimately achieve a form of synthetic immunity that can protect patients, at least those patients who are not capable of forming a protective immune response on their own. And so I think that this adds a third arm to T-cell-based medicines. The most important one is the first one, really, active immunization. This has had the greatest impact, of course, in history. And I sure hope that we'll have a vaccine for COVID-19 sometime. The next one is inhibition of negative regulation with its enormous impact on many cancers, especially those cancers with high mutational burdens. And the third leg here, one, if all of that fails, there's still yet another resort to go and engineer a response and instruct supraphysiological immunity. And there are many people on the slides. There are many visitors to the lab right now, even at this hard time. But I just want to mention my longtime collaborators, Isabelle Rivière and Xian Wang for the CAR T-cell manufacturing and much more at MSK. Rainier Brentians, who was a fellow long ago working on CD19 when everybody told him not to come to my lab because this was crazy stuff. And also Prasad Adusumili, who directs our solid tumor CAR T-cell program at Sloan Kettering. And I'll stop here, and I'm afraid that it's 2 o'clock, Doug.
Not a problem. Thank you, Michelle, for that stunning presentation. Absolutely wonderful. Everyone who is online, we are formally stopping the seminar here. We understand people have appointments and other things they need to do, and we thank everybody who attended. But we will keep the discussion going to address questions that have been raised, and I'll bring those up. So if you can stay with us, we will get to the questions, and we'll ask Michelle to discuss some of these points. So thank you, everybody. Officially ending now, but stick around for the discussion, which should be pretty interesting. I'm going to take these more or less in order that I received them. Michelle, you give us your thoughts on this. So Hadir asked, is there a way to program cancer cells instead of T cells, programming them in a way that caused the T cells to better attack them?
Certainly in a dish, in vitro, or in the laboratory, you can do all kinds of things to tumor cells. And you can increase their ability to be recognized by immune cells. Now, to do that in vivo is obviously much more difficult. Drug delivery, reaching your intended target, the cancer cells in this case, is having said that, there are examples that I think go in the direction of this very good question. There are drugs that you can give, demethylating agents or HDAC inhibitors, for example, that can increase the expression of target antigens. And in doing so, that would create a tumor cell that can be more, potentially anyway, can be more easily recognized. And I do believe that there will be strategies where you will see the combination of a CAR T cell, possibly an antibody for some modulatory function, and also some small molecule or other agent designed to work hand in hand and cooperate with the T cell. Another example of that is the impact of radiation. We actually published a paper a year or 2 years ago showing that if you irradiate a tumor, it becomes more sensitive to CAR T cell activity. And in that instance, we could track that down to an increased to induction of DR5 of a trail receptor. And when we knocked down that trail receptor, we found that that gain in sensitivity of the tumor was lost. So therefore, we think that that contributed to the overall activity of the CAR T cell against those tumors. So that's a second type of combination, which I think it really also should be investigated, radiation plus CAR T cell. If you want to introduce, more complex modifications that require a genetic change in the tumor, people have tried that, and that has proven to be very difficult. Yes, you can do some in vivo genetic modification. It's feasible, but it's rarely efficient. And if your strategy relies or requires that you modify all, or at least very many of those tumor cells, that is a barrier. Now, oncolytic viruses are another way that can operate where they don't need to introduce themselves in each and every tumor cell. They go into some of the cells, they may kill those cells through a cytopathic effect, but then if they do other things that they potentially have to be engineered to do, they too could act on those tumor cells. And the last part of my answer to this question is to use the CAR T cell itself. This allows me to make a very important point. CAR T cells are not just killers of tumor cells. Sure, we want them to kill, to eliminate, to eradicate. But as they go to the tumor, if they go to the tumor, they can be used to release other agents. And there is plenty of literature already on CAR T cells releasing either cytokines or SCFVs or a full antibody or even a bispecific engager, all of which can be released locally as long as the T cell got there. Again, with the goal of mostly in this case modulating the tumor microenvironment, but that could also serve the purpose of modifying more directly the tumor, as Xavier just asked.
Thank you. Galvin asks if you have any insights in the challenges or successes of developing CAR T cells for neuro-oncology applications.
So I would have to refer to the literature here. We have recently started a program, that's a big word, a project on focusing on glioblastoma, but others have been doing that for several years. Ours is to identify targets right now. And so we're this algorithm that I described, relying in large part on cell surface proteomics as well as transcriptomes, is what we're mining at the present to look for more attractive targets than what exists out there at this time. So I don't have as much to say about this as others do who already work on this. But as you may know, there was already a case report. So it's only one case of a glioblastoma treated at the City of Hope, where an adult with a recurrent glioblastoma went into a very profound response for several months, I think for 8 months. following the intratumoral infusion of T cells. And so that gives hope that CAR T cells could work for CNS tumors. Now, of course, many others on the same trial were treated in the same way and did not show those responses. But at least that case, I think, inspires many to pursue this. I certainly see no reason why that could not work, but one has to find the right targets to not damage surrounding normal cells, of course. And there's still a lot to learn about how to make a T cell thrive in the glioblastoma microenvironment.
Tiara Griffin asks, is there a way to determine which patients would be the most susceptible to CAR T toxicities?
We know a few things. In the case of leukemia, for the leukemias in particular, that a high tumor burden, just that, puts you at a higher risk for developing cytokine release syndrome. And so the clinicians are very well aware of that and will keep a closer eye. They keep a close eye on everybody, but a closer eye on someone with a large tumor burden. There are also, the monitoring now is very tight, if I may say, in the first days following CAR T cell infusion. And there are markers that suggest that someone is going to develop a greater CRS. And so in this way, there are ways to be more ready to intervene. But if the question, I think, went beyond that, right? Would there be other markers or genetic factors that you could measure beforehand to know? And I personally don't know of any, but some others may have data on this. We know that rapid proliferation and expansion of the T cells is a contributing factor to these pathologies. And so if, for example, there were a way on some, pre-infusion testing to predict that, there again, you could maybe, again, better anticipate who might be more risk for these toxicities. To me, ultimately, what we need is to, today we have to, recognize them fast and treat them as early as possible. But the real game is to prevent it. And to prevent it means to understand the mechanism. And part of the mechanism is that the rapid expansion is a factor, but it's really the action on the myeloid cells. And so to me, if there are means to mitigate the response of the myeloid cells without compromising the CAR T cell therapy, that we may reach the goal of having, safe administrations, which would then allow these therapies to be really outpatient therapies and, not requiring hospitalizations now and then as they currently do.
Abel has asked a question, and I'm going to slightly rephrase it. What is your experience with repeated boosts of CAR T therapy?
It's somewhat anecdotal. We've done it on I don't know, maybe 10 times or so. And our observation matches what I think others have seen, and this is anecdotal. I don't know if anyone has pulled these data together and published them. If there is such a study reported somewhere, I'm afraid I don't know of it. But anecdotally, what we have observed is that the second infusion tends to not be as effective as the first. You don't induce a complete remission in 90% of those individuals. It's about less than half. So the second infusion tends to be less effective. But I don't really have any further biological insights. One that is clear is that in some patients there is an immune response to the CAR, and there has been from the University of Pennsylvania the case of a dramatic anaphylactic shock on a reinfusion of a CAR T cell with an SCFV that was of murine origin in a solid tumor. And that shows, the potential danger of having someone develop such an antibody response. And that was from Penn, and from the Hutch, Stan Riddell and his colleagues documented some T cell responses directed to the CAR T cell, and they they pinpointed the recognized peptide to some of the fusion points. And if I recall well, it was between 4-1BB and zeta. And so there would be T cell responses that could form. So for that reason, certainly most of us today, I think, would proceed with human SCFVs, at the very least humanized. But then there's still the problem of these junctional peptides, of course, that will come up with any fusion molecule. So anyway, the antibody response is one, but I'm sure then there are many other biological factors why, if the tumor is relapsing, why it may often not respond so well to a second infusion. It's a great question, and we need to, the field needs to look at that. Should you come back with a different CAR T cell or modify your CAR T cell to preempt or treat this scenario?
Sean Murphy asks about studies that combine CAR T therapy with immune checkpoint blockade.
Yes. We think that they're very interesting. We have a publication on this, by the way, where we show that CAR T cells targeting mesothelin in a solid tumor model, a model of pleural mesothelioma, that these T cells, when they're infused at low dose, I insist on giving relatively low doses to mice because I fear that when you give lots of T cells, things work well, everybody's happy, but it doesn't really recapitulate a real situation. Whereas when you give lower T cell doses, you're really forcing your T cells to go to work. They have to engage antigen, they have to expand, they have to maintain their function over time, and hopefully not undergo T cell exhaustion or apoptosis, or at least not do that too early, so as to have the time to have a meaningful response. So in that setting, Prasad Ajasumili, whom I mentioned earlier, had noted characteristics of T cell exhaustion in those CAR T cells. When he treated those mice with a PD-1 blocker, he could restore responses in those mice. And so that has informed a clinical trial performed at Sloan Kettering in patients with malignant pleural mesothelioma with infusion of T cells in the pleura, as had been demonstrated in the mouse models, and with subsequent infusion of pembrolizumab in those patients. And it's a phase 1 trial, so I can't. We don't have too many mechanistic insights, and there's no control arm to the study per se. But we believe that this enhances the therapy. In the mouse models, it was very clear, and so to just keep it simple, to me, it is very much worth investigating.
Fantastic. By the way, there are still a few more questions. I won't wear you out too much, but there are lots of thank yous and congratulations as well for a wonderful presentation. Kenneth Caldwell has asked 2 questions. This one is fairly specific, and then one that is more general. Some recent papers suggest CD3 epsilon may play an important role and should be included in CAR T cells along with CD3 zeta. Do you think this may be true, and is it a promising future technique?
I think it's possible, but we haven't explored that. I've seen some of these recent studies, so all I can say is that it looks promising, but we haven't done that.
And do you believe that allogeneic off-the-shelf CAR T cells are the future? And if so, what strategy seems most promising?
Well, it's a big question. Obviously, from a point of view of drug delivery, distribution, and cost of the therapy, above all, it would be enormously desirable to have so-called off-the-shelf therapies. They don't have to be, quote, universal, which I think is a term that should not be used as, there isn't one cell that you can give to all of humanity, but if there were ways to make batches of cells that could be, designed for a specific indication and provided to recipients, perhaps with a partial HLA match or some other form of conditioning that doesn't exist yet that could temporarily block the rejection of those cells, that yes, we maybe could have allogeneic solutions down the road. But it's not a simple simple task to fulfill. You can prevent the donor cell, I'm sorry, from reacting against the host and triggering something like a graft versus host disease. If you use virus-specific T cells that are not alloreactive, if you knock out the T cell receptor using a targeted nuclease, or if you use NK cells or invariant NK T cells or gamma delta T cells that are known not to cause graft versus host disease, you could mitigate that problem and just have the CAR or combination of CARs direct the function of the T cell towards the tumor. But then the other problem is that your recipient will reject these cells. And I think the world awaits to see some solutions to that. There are a bunch of ideas that are bounced around, but to my knowledge, they haven't yet been demonstrated yet to work outside of very specific settings, particularly in a post-transplant setting, which is a particular setting where the recipient is relatively immunocompromised. But if you're thinking about other cancer patients who are not, who still have an immune system and whom you don't want to endanger with some, non-specific deep immunosuppression, then we need to come up with really more creative solutions. And in that regards, I think that the gene editing that I mentioned will be very helpful. Perhaps producing the cells from even their stem cell reservoirs may in some ways facilitate that task. But above all, it's a big biological challenge, right? It's you don't need to induce perhaps the deep tolerance that you would need to induce for an allogeneic organ transplant, but you still need allogeneic cells functioning in a recipient's fighting their cancer without being rejected for a certain amount of time. How do we do that? I hope that some of our listeners who are maybe thinking about working in this field will come up with the solution.
Couple more questions. What are the main obstacles, in your view, to developing an effective CAR T for AML?
I think AML is challenging. Everything's challenging. All cancers are challenging. But if you contrast it to the B-cell malignancies or myeloma or T-cell leukemias or lymphomas, in all of those, you can find targets that are not shared with hematopoietic stem cells. But the challenge of AML is that leukemia stem cells share many features with active hematopoietic stem cells or early myeloid progenitors. And so the challenge, I believe, is how to design a smarter CAR T cell that will be able to presumably target a molecule that could be also found on the normal hematopoietic stem cells, but spare those normal hematopoietic stem cells. If not, you're going to need a bone marrow transplant to rescue. Now, curing these patients is a big challenge, and so I'm not saying there would be anything wrong with that, of course. But if the question is how could we come up with a CAR therapy that is not a bridge to transplant or does not obligatorily require a transplant, then that question, that project, that challenge is how do you get to LSC, leukemia stem cells, but not normal hematopoiesis? I think there are, I hope anyway, that there are solutions. I think they require a more sophisticated and clever strategy than what was sufficient, for example, for B-cell malignancies.
There's three more questions, so I'll ask them individually and take them as you wish. This is from Michael Schwab, who asks, are different glycosylation patterns between cancerous and healthy cells being considered as potential targets?
Yes, and thank you for asking the question. I think that glycolipids and carbohydrates could make very good CAR targets. And definitely there are attempts there are CAR T cells that have been specific for, sialyl Lewis A or glycosylated glycosylated forms of MUC proteins, and GD2, which is a ganglioside, also is a potential target for CAR T cells. And so I think that's an interesting aspect of CAR T cells, and it should be explored. It's not easy to find tumor-specific targets outside of the world of mutations and neoantigens, and so such targets, I think, could be of great interest. They're harder to study, right? Your transcriptomes are not going to help. And unfortunately, we have tons of transcriptomes, but they're not informative. As I mentioned, we rely increasingly, when we can anyway, on cell surface proteomics, although that too would not capture these. You need glycomics and other explorations to identify such targets. But anyway, my bet is that there are some great targets to be discovered in that space.
That's fantastic. Two more questions. Manfred Lehner asks a great question. Which strategies do you believe have the greatest potential to improve the therapeutic window of CAR T cells with regard to on-target, off-tumor toxicity?
Ooh, that's a very elaborate question, actually. I think that if I rephrase the question, it's again about dealing with the challenge that a target is on tumor cells, but some normal cells. And again, the CD19. So long ago, we thought CD19 was okay because the B-cell aplasia, not ideal, is nonetheless tolerable. And we agree that that's not the case for a lot of the other antigens. We can't target hepatocytes or normal kidney, and so this is one of the central questions in how do you either you find those molecules that are really tumor specific. Again, that's the best scenario, or because there aren't so many of those, we have to come up with creative ways, walkarounds, to somehow restrict the ability of the T cell recognizing antigen X to only recognize antigen X, on the tumor, and not recognize antigen X when it's on another cell type or in another location. And so then we get into, how to block the activity against the non-tumor cell. There are inhibitory receptors that have been proposed, ICARs, actually based on PD-1 fusions in particular. And then there are other strategies to conditionally express a receptor that, again, that's at risk of causing a toxicity such that it would only be expressed transiently when the T cell is in the tumor. So the T cell has to know that it's in the tumor. So it has to react to something, either an antigen or some condition that is particular to the tumor to induce the expression of this molecule. There's this sin notch model that I'm sure the person asking the question is well aware of. That's maybe relevant to this setting. And then there are a combination of targeting and signaling approaches that I think also aim to tackle this challenge. I believe that there will be solutions to that. They're just more sophisticated than, putting a CAR in a T cell and letting it go. That worked for CD19 CAR therapy, but now we need more sophisticated things.
And the last question, and I will just read it to you because this is outside my expertise. This is from John Lucas, who asks, would use of localized RT pre-CAR T infusion limit ICANN-related side effects due to suppression of immune-related inflammation from myeloid suppression locally? I'm thinking CNS tumors in tough spots where ICANN could lead to serious side effects.
Yeah, that's a great question. I don't have the answer to it, but I'm sure you can guess that I completely agree with the question. So, CRS is in part, I think in large part, the severity of CRS is in large part determined by the number of myeloid cells that are in the immediate vicinity of the tumor cells and the CAR T cells. There's always macrophage in the tumors, but some have many more. They're also recruited once the CAR T cells are activated locally, and they come in very fast, and they don't just accumulate, they're also activated by the CAR T cells. We showed the CD40 ligand played a role, but there are DAMPs and many other things that presumably contribute to this local activation. So, we showed that IL-1 receptor antagonist can reduce that. But I'm sure that there would be many other ways, presumably anyway, to either mitigate or limit the recruitment of the macrophage, or if they're already there, at least to limit their activation. And I agree that the radiation question is worth asking. I wish we had done that experiment already, but we didn't yet.
The very last question is going to come from me on in this regard. Lou Stout at the NIH demonstrated some time ago that BTK, one of the important kinases in B-cell lymphomas, which is targeted with a specific drug and is very effective, is expressed in inflammatory macrophages and drives the production of IL-6. And he's had some remarkable results with the BTK inhibitor, which is extremely well tolerated, in suppressing IL-6 responses from macrophages. Has anybody combined CAR T with a BTK inhibitor?
Well, to study its effect on IL-6 production by myeloid cells, not to my knowledge. And what I do, so the dasatinib has been tested, so it's a different, I'm not following up on your question here per se.
Right.
And has been shown to inhibit CAR T cell activation and very dramatically and rapidly arrest their expansion, and also in a reversible way once you stop giving the drug. And so I think that that's one precedent where a rapid intervention with a small molecule has been shown to be very quickly active. But I do not have an answer to your question, but I do believe that it's a very interesting one, and that how to block this axis IL-1, IL-6, it appears so far that blocking it does not come at the cost of interfering with the therapy.
Right.
And so therefore, I think it is really worth further exploring, because, again, it would, of course, be better for the patient not to undergo these toxicities. But I'm thinking of the whole logistics here, having the patient near the hospital, or even having them at St. Jude when they receive their CAR therapy. In the event that there will be such a toxicity, if there were ways to simply and effectively intervene very fast, this therapy could really become much more of an outpatient therapy.
That's fantastic. I'm just going to note, Michelle, that there are still well over 100 people with us.
Okay.
You had more than 460 attending your talk. I think this is a smashing success. Michelle, once again, thank you for this. It was absolutely fantastic. Thank everybody. Thank you for fantastic questions. I think
Yes.
the discussion was as informative as the amazing talk. And I hope you'll all join us for the next one of these, which also promises to be very exciting. We're going to have these continuing for quite a while. This was the inaugural one, and I think it's a great way to start. Congratulations, Michelle, and please keep up the good work.
Pleasure and honor are mine.
It's so great to see you. So we'll officially end this all, and thanks for joining us. Bye-bye, Michelle.
Bye-bye.
Bye.